Good morning. It's October 31st, and this is your daily brief in science. Here's everything you need to know. A recent study from Mount Sinai has unveiled a crucial connection between heart attacks and increased sleep, revealing that this additional rest can aid in heart healing and reduce inflammation. The research monitored over 80 patients in the 4 weeks following their heart attacks, finding that those who experienced poor sleep had a worse prognosis. Specifically, patients with inadequate sleep post-heart attack faced twice the risk of subsequent cardiovascular events, highlighting the critical role sleep plays in recovery. Published in the journal Nature on October 30, 2024, this comprehensive study involved both mice and human subjects, showcasing the depth of the research. Funded by multiple National Institutes of Health grants, the study underscores its significance in cardiovascular research. Mount Sinai Health System, known for its leadership in cardiology and heart surgery, consistently ranks highly in national and global assessments. The findings illustrate how the heart and brain communicate through the immune system to facilitate recovery after a cardiovascular event. The research, led by Huyn et al., emphasizes the importance of prioritizing sleep in clinical treatment following heart attacks, especially in intensive care units. Additional studies on healthy adults also indicated that sleep restriction triggers sympathetic stress and inflammatory responses similar to those seen in heart attack models. Sleep is essential for tissue and cell repair, playing a significant role in reducing inflammation throughout the body. Moreover, blocking the accumulation of monocytes in the brain was shown to prevent the increase in slow-wave sleep after heart attacks, further supporting the connection between sleep and cardiac health. Researchers at the University of Tokyo have made a groundbreaking discovery that could change our understanding of cellular biology. They successfully inserted chloroplasts from red algae into cultured hamster cells, allowing these animal cells to photosynthesize light. This innovative technique has shown that the photosynthetic activity in the implanted chloroplasts lasted for at least 2 days, indicating a significant advancement in hybrid cellular technology. This research challenges the long-held belief that animal cells would digest chloroplasts before they could function. Evidence of ongoing photosynthetic activity and the presence of chlorophyll in the hamster cells supports this new understanding. Led by Professor Sachihiro Matsunaga, the team is the first to report photosynthetic electron transport in chloroplasts implanted in animal cells. Interestingly, the hamster cells that were cultured with the chloroplasts exhibited increased growth rates. This suggests that the chloroplasts may provide a new carbon source for the host cells, potentially contributing to a carbon-neutral society. Professor Matsunaga envisions the development of planimal cells that integrate both plant and animal characteristics. The researchers are exploring how chloroplasts and host cells may exchange nutrients, which could lead to various biotechnological applications. This research has potential implications for growing organs and tissues for transplants, as well as in the production of lab-grown meat. Advanced imaging techniques, including confocal and electron microscopy, were employed to monitor structural changes and verify photosynthetic function in the modified cells. Using laser light and pulse amplitude modulation fluorometry, the team confirmed that the implanted chloroplasts were actively performing photosynthesis. Their findings were published in the Proceedings of the Japan Academy and received support from various Japanese science grants, marking a significant step forward in the field of hybrid cellular technology. Recent research has ignited excitement in the scientific community regarding Miranda, a moon of Uranus, as it may possess a subsurface ocean that could potentially support alien life. Conducted by scientists from Johns Hopkins University and the University of North Dakota, this study was published in the Planetary Science Journal, presenting groundbreaking findings on this intriguing celestial body. The research indicates that while a deep ocean likely no longer exists, liquid water could still be present beneath Miranda's icy crust. The study suggests that an ancient subsurface ocean may have comprised nearly half of Miranda's total volume, a significant comparison to Earth's Mariana Trench. Notably, the absence of surface cracks, which would typically indicate expansion from a frozen ocean, supports the hypothesis of a subsurface ocean. The team believes that Miranda's interior may still be warm enough to harbor a subsurface ocean today, although further data is required for confirmation. Researchers propose that Miranda has likely moved past its past orbital resonance, leading to a cooling interior, yet the possibility of an ocean existing beneath the surface remains. Caleb Strom, one of the researchers, emphasized the importance of understanding the conditions that allow icy moons to be classified as ocean worlds. Tom Nordheim, a co-author of the study, expressed surprise at the potential discovery of an ocean in such a small moon, highlighting its unique geological features, including a heavily grooved and cratered surface influenced by tidal forces. Future missions, such as JUICE and Europa Clipper, may enhance our understanding of Miranda's subsurface environment, even though they primarily focus on Jupiter's moons. Historically, scientists believed Miranda would be a frozen sphere due to its small size and age, but recent discoveries about icy moons have challenged this long-standing assumption. A new study sheds light on the relationship between body mass index, or BMI, and post-acute sequelae of SARS-CoV-2 infection, commonly referred to as PASC. Conducted by Dr. Ting Zhou and his team at the University of Pennsylvania, the research analyzed data from 172,136 children and young adults across 26 children's hospitals in the U.S. The findings, published in JAMA Network Open, reveal that higher BMI is associated with an increased risk of developing PASC. Specifically, the study indicates that obesity raises this risk by 25.4%, while severe obesity escalates it by 42.1%. Among the participants, nearly half, or 49.7%, were classified as having obesity or severe obesity. During the follow-up period, 0.8% were diagnosed with PASC, and 26.4% reported at least one symptom related to PASC. The average age of participants was 12.6 years, with a demographic breakdown showing 52.4% were female and 50.7% identified as non-Hispanic white. Notably, the significant association between BM1 and PASC was primarily observed in non-Hispanic white participants. PASC can impact various organ systems, with its prevalence ranging from 1.6% to 70%, indicating a pressing need for more research into its causes, particularly among children with obesity. The symptoms of PASC are diverse, including abdominal pain, acute kidney injury, cognitive dysfunction, and heart disease. Previous studies have largely focused on adults, leaving a gap in understanding how obesity affects PASC risks in children. The authors of the study highlight the necessity for targeted research to identify specific PASC symptoms linked to different BMI levels, as well as the importance of healthcare interventions for at-risk youth. They advocate for managing obesity as a modifiable risk factor to reduce PASC risks and improve overall health outcomes, emphasizing the need for public health initiatives that encourage healthier lifestyles. A recent study published in the American Journal of Psychiatry on October 30, 2024, has the potential to change the landscape of treatments for anxiety and depression, which affect millions of people worldwide. The research focuses on intercalated cells in the amygdala that express the FOXP2 gene, a finding that could lead to targeted therapies for anxiety disorders due to its role in regulating signal traffic within the brain. These FOXP2-expressing cells also showed expression of the neuropeptide FF receptor 2, or NPFFR2, suggesting that drugs aimed at this receptor might provide new avenues for treating anxiety-related conditions. The study, led by graduate student Sean Cambodge, utilized single-cell RNA sequencing to analyze the cell types present in the amygdala of both humans and rhesus macaques. This analysis revealed important similarities and differences in amygdala cell types between humans and non-human primates, which is essential for translating findings from animal studies to human applications. The research team categorized cells based on active gene expression patterns, a method that may yield insights into the underlying mechanisms of emotional disorders. Drew Fox, an associate professor at UC Davis and the study's senior author, noted that traditional measures of amygdala size and structure are not reliable indicators of emotional disorders. He emphasized the amygdala's crucial role in processing emotions, particularly fear and anxiety, and highlighted that understanding specific cell types could unveil choke points for developing more effective therapies. The research was supported by the California National Primate Research Center, the NIH, and the Simons Foundation, showcasing a collaborative effort to map cell types in the amygdala. This work is pivotal for enhancing our understanding of emotional processing and its implications for mental health. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.